Hi everybody, and welcome to the latest VodCast for Honors Biology at Desert Ridge High School. I'm Mr. Galladay, and today we're going to be talking about DNA. And in particular, what we want to talk about DNA is to try to understand how it is that we know what we know. You've undoubtedly heard something about DNA in the past. You probably know that it has this double helix shape, of course, which is kind of famous. But on the other hand, if you've ever looked at DNA, if you've ever extracted DNA from a strawberry or a kiwi fruit, or seen your own DNA, you know that you can't see this double helix structure with your bare eyes. This is not something that's visible to us, and DNA is not very interesting looking on its own. And so the question may have entered your mind is, well, if this stuff is so important and is the blueprint of life, why does it look like snot? And you know, you're probably not the first person to wonder this, and so that's what I'm going to try to answer today is a little bit about the background of how we know what we know, and how did we get to this point where we have this knowledge today. Okay, so this is a good point to update your table of contents, update the organization in your notebook, and as always, you can pause or rewind the recording if you miss something or want to get it in a little more detail. Okay, we're going to be looking at this as sort of a scientific mystery, and the question that we're trying to answer is, what are genes and what are they made of? And so this is something that around the time of the mid 1800s, early 1900s, that people began to ask in a formal scientific way, and so we're going to see how we came to that knowledge. Okay, before Mendel there were geneticists. Only they didn't call themselves geneticists at that time. They called themselves farmers. Farmers were, of course, interested in having cows that grew fatter. They were interested in having horses that would run faster or haul heavier loads. They wanted to grow crops that were able to produce more grain or more fruit and feed more people. And so people had been doing genetics experiments for thousands and thousands of years, long before there was any formal science of genetics. So at that time, people just assumed that all inherited traits were basically a blending of the parent traits. In other words, if you had one very tall corn plant and a very short corn plant, that the offspring would be sort of a medium-sized corn plant. And that of course is true for many traits, but as Mendel showed, certainly not true for all observable traits. Okay, and so the things that people knew, or the things that people thought they knew, in other words, the rules of genetics, were sort of a mixture of personal experience, personal observation, some guesses, and just some outright superstition. Until Mendel came along, no one had really treated this in a very scientific manner. Well, of course, the guy who changed our thoughts about genetics, or one of the important people who changed how we studied genetics, was Gregor Mendel. He was a monk who lived in a monastery in what is now Czechoslovakia, or part of, which at the time was part of Austria, the Austrian Empire. And he worked with pea plants over many years and grew thousands and thousands of pea plants and collected thousands and thousands of pages of data over a period of several years. Now, he was really the first to use data and statistics to study inheritance. In other words, he did not rely on sort of the conventional wisdom of the day, but tried to understand this on his own, using data and statistics. And so what he developed, of course, were probabilities of inheriting various traits. and we've come to know those as Mendel's rules or Mendel's laws of inheritance. Now, one thing that's kind of interesting to think about Mendel is what did he know about DNA? What did he know about chromosomes? And the answer to that, of course, is he knew absolutely nothing about those. It wasn't until much later that that people connected his ideas with chromosomes and DNA, and that's what we're going to see here today. Okay, working around the same time, Gregor Mendel published his work in 1866, and another fellow who was also working in Western Europe was a guy named Friedrich Miescher. Now, Miescher had started off his career as a physician and later became interested in chemistry and biochemistry in particular, and he worked as a physician in actually during part of the Crimean War, and was interested in infections and what it was that sometimes kept people from getting infected and protected them from infections. Now, the area that he was studying was actually the white blood cells, or the nucleus of white blood cells. And the way he collected this material was actually from the pus that was secreted from various wounds, and he collected it from old bandages, which, if you think about it, is kind of a disgusting way to do research, but that was where his curiosity took him, and so what he was able to do was to isolate these white blood cell nuclei, and he found a new substance on there that he realized was not a carbohydrate, it was not a protein, and it was not a lipid. It was some entirely new substance that he called nuclein. Nuclein, long after Miescher was dead, we began to call by the name of deoxyribose nucleic acid. But he was the first person to realize that there was this new substance that was not a protein, not a carbohydrate, and not a lipid. Now, one question to ponder here is, how would Miescher know that this was something that was actually new? And the clue to that was that it was none of the other three biological macromolecules. And how would he know that? Well, if you think that he was a chemist, and you think back to what we learned earlier about these macromolecules, and you might remember that nucleic acids such as DNA contain the element phosphorus, which is not found in proteins, not found in lipids, and not found in carbohydrates. So that's how he was certain that whatever this nuclein stuff was, it was something new. Now, one thing that Miescher and Mendel have in common is that their work was, at the time, not thought of as very important. They published their work, and then for the most part, they were forgotten. Nobody paid much attention to it at the time, and it wasn't until really 50 years or so after this that people began to look at this stuff and realize that there was something important about it. One of the first people to begin to combine the work of these two guys was a couple of scientists that worked on the eastern part of the U.S. Named Nettie and Edmund Wilson. Now, they worked independently of each other. They were sometimes they collaborated, sometimes they were competitors, and this was in the very early 1900s. And you see here these are chromosomes of fruit flies, and fruit flies are one of the organisms that they worked with, but they worked with a variety of insects. And the thing that they learned was that male and female insects have different chromosomes, which of course you probably know. Males have females rather have two X chromosomes. Males have an X and a Y chromosome. But they were the first people to discover this, or to notice this. And they noticed it, and they carried it even to the point where they realized that they could determine the future sex of an insect before it had developed. So in other words, they could look at the embryo cells and look at the chromosomes in those cells, and they could determine whether or not that was going to become a male or a female insect. Okay, now you might wonder, well, what does this have to do with genetics, and why is this important? And of course, the answer to that is that they were the first people to realize that whatever a gene is, or what Gregor Mendel called these inherited factors, they must be on chromosomes, because chromosomes had something to do with traits. And so whatever a gene was, it had something to do with chromosomes. A colleague of both Nettie and Edmund at the time, who worked just a little bit later, but also in the early 1900s, was a gentleman who we remember as sort of the fruit fly scientist. His name was Thomas Hunt Morgan. Very famous scientist, and did lots and lots of experiments with fruit flies. Did thousands and thousands of experiments with fruit flies, and what he was able to learn is that each gene, in other words, each trait had a gene which was at a particular location on a chromosome. Okay, he was able to deduce this by doing all these thousands of experiments, and noting changes that were surprising to him, noting changes that were unexpected, and the frequency with which those happened. Okay, so at this point we know quite a bit, but there's an awful lot we don't know, so we're going to take just a minute and sort of ponder all this stuff. Okay, at this point, the idea of a gene, or the word gene, was beginning to be used, but nobody knew exactly what a gene was. All we knew was that a gene was some sort of a factor that caused an inheritable trait. Okay, so if we talk about a gene for eye color, then it was just sort of some abstract idea that had something to do with causing this trait, but we didn't know anything else about it. We knew that there were chromosomes, we knew that there were proteins, we knew that there was DNA, we knew about all these things, and we knew that chromosomes had something to do with whatever a gene was. It was probably on a chromosome. Now, when people studied chromosomes, when they took chromosomes and analyzed them, one of the things that right away they found was that chromosomes are made of both DNA and protein. Okay, so you've got this funny looking thing called a chromosome. You know that this funny looking thing has something to do with inheritance, and you know that this thing has both something, this DNA or this nuclein stuff, and you know that it also has some proteins. Now, what people knew about proteins is that they were very, very complicated. They knew that there were 21 different amino acids, and they knew that DNA was a pretty simple substance. There were only 4 nucleotides: adenine, thymine, cytosine, and guanine, and they knew that they were present in roughly 25% each. So they didn't seem very interesting. So we knew that proteins were very complicated. We knew that DNA was very simple. So you might think of it this way: if you were looking for something very grand and very important, like the blueprint for life, would you look at something simple like DNA, or would you look at something complex like proteins? Well, most people at the time thought, well, it must be a protein. Okay, so this is where we're going to hold up this VodCast, and we're going to continue this on the next one. And for Desert Ridge High School in Honors Biology, I'm Mr. Galladay, and I hope you have a good day.